So in this video, we're going to have a look at the digestive process in the stomach. Now, the process is carried out by the stomach, and we do it in a few ways. So let's talk a bit about what the function of the stomach is. So it carries out the breakdown of food. It also serves as a holding area for the food. And of course, once you break this down, it's going to make a material that we're going to call chyme. And that's going to be dropped off or delivered to the small intestine. The other thing is that we're going to produce hydrochloric acid, which will denature the proteins that are in the food while in the stomach. And we also have some enzymes that are released here, and we talked about those in class. We have pepsin, which carries out the enzymatic digestion of proteins, such as the milk protein casein is broken down by renin in infants, and that's another one we could look at. Lipid-soluble alcohol and aspirin are absorbed into the blood here. So we typically don't think of the stomach as actually absorbing stuff, but it does. And really the two examples would be a lipid-soluble alcohol and aspirin. Now, there's only one function that is absolutely essential to life and when it comes to the stomach, and that is intrinsic factor. And we need intrinsic factor for the production of vitamin B12 absorption. And of course, vitamin B12 is required for red blood cells to mature. And if you do not have intrinsic factor or enough of it, you will end up with a condition called pernicious anemia. Now, this can be overcome by using injections of B12. So let's have a look at gastric secretions. Now, you make about 3 liters of gastric juice a day, and it's regulated by a number of factors. The two main ones are neural mechanisms and hormonal mechanisms. Now, not surprisingly, the vagus nerve stimulation increases secretion, and remember that's a parasympathetic effect, and the sympathetic stimulation decreases secretion. Now, when we look at hormonal mechanisms, we can see gastrin stimulates hydrochloric acid secretion by the stomach. And also gastrin is an antagonist to hormones that we find in the small intestine. Now, when we look at it, there are 3 phases of gastric secretion. We have the cephalic, we have the gastric, and the intestinal phase. So all 3 of those are part of the gastric secretion. Now, let's look at them individually. So the cephalic, this is a reflex phase, and this is triggered by external things such as aroma, smell, taste, thought of food. It's sort of that Pavlovian type of response. Next thing is that we have the gastric phase, which lasts for 3 to 4 hours and provides about 2/3 of the gastric juice released. Stimulation of the gastric phase, we get distension of stretch receptors, which initiates both those long and short reflexes that we make reference to earlier in class. Also chemical stimuli, partially digested proteins, caffeine, low acidity, all of these activated enteroendocrine G cells. They get stimulated and they will secrete gastrin. Now, the release of gastrin initiates hydrochloric acid release from the parietal cells. And of course, this is going to have a role in activating enzymes. So parietal cells do secrete the hydrochloric acid. So we get binding of receptors on the parietal cells and stimulating of enteroendocrine cells to release things like histamine also. We have a buffering action of ingested proteins, causes in pH to rise, which activates more gastrin secretion. So and that was basically what stimulates gastric phase. Now, inhibition of the gastric phase would be low pH. That inhibits gastrin, and it occurs between meals, and it makes sense because you don't want to be producing all these enzymes, at least hydrochloric acid, between meals because it's very risky for the actual tissue of the stomach to have this stuff there all the time. Occurs during digestion as a negative feedback mechanism. The more protein, the more hydrochloric acid is secreted, causing a decline in pH, which inhibits gastrin secretion. Now let's quickly look at the intestinal phase. Now it begins with a brief stimulatory component followed by inhibition. Because once again, we don't want to turn these things on for too long. We want short bursts of activity, get the job done, finish with the job, downregulate. So stimulation of the intestinal phase. So partly digested food will now enter the small intestine and causes a brief rise in the intestinal or the enteric gastrin. And this encourages the gastric glands of the stomach to continue their secretion. Stimulatory effect, as I said, is brief and is overridden by the inhibitory stimuli as the intestine fills. And this makes sense because you don't want to send too much from the stomach to the intestine at one time. You want to work on the material in small little chunks so you can actually deal with it properly. Inhibition of here, what can inhibit the intestinal phase? There's 4 main factors in the duodenum that cause inhibition of the gastric secretions. So we have, first of all, we have distension of the duodenum because the chyme is entering, so we're going to get stretching of it. Presence of the acidic chyme. Of course, the chyme is just coming from the stomach, going in the small intestine. It'll be acidic. The presence of fatty chyme. We'll have some fatty material that was in whatever we ate. Its presence will have an effect. And the presence of hypertonic chyme will also have a role in inhibition of the intestinal phase. So 4 factors that lead to this. Now, as I said, the inhibitory effects protect the intestine from being overwhelmed by too much chyme or acidity at once. The small intestine doesn't have the same protective mechanisms as the stomach when it comes to acidic material, so you really have to only let a little bit of material in at once and deal with it, and then a little bit more later on. Inhibition is achieved in 2 ways, the enterogastric reflex and basically the enterogastrones. So let's have a look at those 2 things right now. Let's look at the enterogastric reflex. So the duodenum inhibits acid secretion by the stomach, and this is through the enteric nervous system, which we talked about in class, and that's through the short reflexes. The other thing that has an effect would be the sympathetic nervous system and the vagus nerve via the long reflexes. So there's a couple ways we can start to modify this. We also have the enterogastrones, and these are duodenal endocrine cells, and they release 2 important hormones that are going to, let's underline, inhibit gastric secretion. And these are secretin and CCK, which stands for cholecystokinin. Now, your book has a really good diagram that sort of walks you through all of this. So once you've gone through the video, once you've looked at your notes, go back to this diagram and try and work your way through. It really summarizes it nicely.